Good evening, everyone. Welcome to the first Kelvin Club talk of the term. So tonight we have Dr. Alex Borodavka. Dr. Borodavka got his bachelor's and master's at the Kiev National University. Following a PhD at Leeds, he received a Sir Henry Dale Wellcome Fellowship to develop fluorescent techniques to investigate RNA viruses with segmented genomes. He founded his group in 2019 and recently was appointed assistant professor at the Department of Chemical Engineering and Biotechnology. So without further ado, I'll let you take over.
Thank you so much, Margaret, and thank you so much for staying up late despite dinner, wining and dining. It's my great pleasure to be in this beautiful lecture hall as well. And I appreciate that a lot of you are probably not virologists, not biophysicists or RNA biologists. You're probably kind of starting off in your research career, some of you. So I'm going to talk a little bit about some of the pathway that set me up and what we're doing now, my group currently does with viruses with segmented genomes, as Margaret pointed out. And I also should say, not only today I realized, actually Margaret and I, we published a paper together, so earlier, a few weeks ago, literally, and also was lecturing Margaret last year, as it turns out. So, okay, I'm going to talk about something which is super topical and hot now, which is RNA, because the recent Nobel Prize went to, say, mRNA vaccines, and I think it's a really amazing time to be alive and to work on RNA. Really, for me, it's a great privilege. And not only that, it's also obviously the viral RNA that kind of put RNA in the spotlight, and we really see this huge transition. We see a lot of companies now popping up, a lot of companies with interest in RNA therapeutics. Some of you who do dabble biochemistry or maybe physics or chemistry or somewhere interface all these disciplines, we're in a very powerful position now, and hopefully you will also discover yourself in this different area of research where you can contribute to something really important. So, viruses. Why did I choose to work on viruses? Well, first of all, I think I was having a conversation at dinner tonight with Graham Christie here, who's a microbiologist, and I worked on viruses, but both are kind of equally fascinating, microbes, viruses. They're a major source of discoveries in molecular biology. If you think about it, okay, so there are very smart people who worked on viruses, usually physicists, chemists, not even biologists, because these kind of really simple questions physicists would ask about viruses, self-assembling systems that can replicate themselves and reproduce and multiply in huge numbers within a few hours post-infection, especially with bacteriophages that infect bacteria. And so, just a few examples. If you're kind of interested in the history of molecular biology, you realize quickly that all these big discoveries, these big prizes that went to this, the titans of molecular biology, they were pretty much somehow somewhat related to virus research. So if you look at all these Nobel Prize winners here, and the last ones, even the latest one, which I didn't include, this slide, I put it together probably just before the latest prize was announced. But you can see Jennifer Doudna, who's coming to Cambridge next month. She's giving a talk, for, again, talking about CRISPR, genome editing techniques. This was all somehow related to virus research, right? So I guess what I want to say, if you study viruses, your name could be next on this list, hopefully. I don't think my name's going to be on this list, but I still like working on the systems because they're really fascinating. And the reason why they're fascinating is because A, there is huge diversity. This is just kind of a snapshot of the viruses that we know at the moment, and you can see how many species they are. And they're all different. They have different morphologies, they have different shapes, they're highly symmetrical, they're beautiful, they fascinate mathematicians, they fascinate physicists, but also they're really important for us because they infect plants, they infect bacterial cultures. People who like yogurt and things, they get infected by bacteriophages. And then they obviously kill animals, and then they kill humans. So we really want to understand how to make these things do something good for us, essentially, and also know how to control viral infections. One fascinating aspect of viruses is that they're essentially kind of spring-loaded nanocontainers. And this slide I use when I teach virology in Department of Biochemistry. I always tell my students they're kind of metastable system. What that means is that they kind of spring-loaded nanocontainers that contain genetic material. It could be RNA or DNA, and they have to be robust enough to protect that genetic material because they need to deliver that cargo, that payload into cells, but they can't be too stable, because if they're too stable, they won't be able to let go of that thing. So they have to kind of sense the environment and know now is the time to pop up and just release that nucleic acid, release that cargo into cell, or actually now it's not the time and you kind of want to keep it all together. So one of the classical studies on virus assembly were done on very simple viruses, and this is plant virus, a classical virus called tobacco mosaic virus. This is just kind of intro 101 on viruses I'm giving you, and kind of set the scene. And essentially with this virus, tobacco mosaic virus, it was a really important pathogen, one of the first viruses ever described. So several virologists, Ivanovsky, who was a microbiologist, he was studying plant diseases, and it was really important because tobacco industry was a huge thing, obviously, and they spotted that tobacco plants were getting sick, it would affect their crops, and they started investigating what the agent of this disease was, and they quickly realized that there was tiny agents that would infect plants which would filter through bacterial filters, and so they called it filtrating agents. They didn't know what virus is. A virus from Greek, poison, that's essentially what virus means. And so this was one of the first viruses ever discovered by Ivanovsky. We're talking about 1907, 1905. So, and then subsequently became such a great model system for studying this general self-assembly principles that Richard Parham, who was in Cambridge, he studied extensively how these viruses recognize genetic material, nucleic acid, and how it assembles this beautiful kind of rings around this nucleic acid to protect it, to form pretty much beautiful rods that would protect this virus, protect the genetic material of this virus. So this is actually one of the oldest papers in 1975 from Richard Parham's group, by Richard Parham himself. And so essentially what several groups they showed with this virus is that you can pretty much disrupt this virus. You can treat it really harshly with several chemicals and then disrupt it, take it apart, and then if you allow those things to interact with each other after they've been renatured, so you put them into kind of quasi-native state, it can easily self-assemble. It can easily reform these infectious particles again, which was really fascinating. And it turns out it's not the only virus that can do it. There are other simpler viruses. Now I'm getting into my PhD times when I was working on a slightly different virus, and this is bacteriophage. Now this virus infects bacteria. This virus infects E. coli. And essentially my PhD was trying to understand what imparts specificity. So in a cell, you have multiple viral proteins, you have viral RNA, but you also have other non-viral RNAs, and the virus needs to recognize itself cognate RNA versus non-cognate RNA. And it's a highly efficient process, otherwise viruses wouldn't just simply replicate. So that was essentially the gist of my PhD, trying to work out where the specificity comes from in this RNA protein recognition. And so we did a bit of biophysics, we did a bit of RNA biology, and we know about this phage that within this particular viral genomic RNA, there is a small stem loop, which is only 90 nucleotides, that is recognized by its cognate viral protein. So the viral protein evolved to recognize this RNA with really high, well, I'm going to call it affinity, so it recognizes with and tightly binds. And that binding event is really important for pretty much the entire life cycle of this virus. It's also really important for recognizing itself, its own RNA versus non-viral RNA, and then packaging that RNA that gets inside this particle and kind of assembles around this nucleic acid. So we published this paper where essentially we said these cognate RNAs are recognized by the proteins, but the assembly of this multiple subunits, viral subunits, is kind of encoded in this protein-protein interaction. So multiple cold protein subunits will collapse around the nucleic acid, around its own RNA, and each RNA kind of evolved a mechanism of this bringing multiple subunits together. So I kind of brought this prop. I'm not sure this is going to work, but I'm going to try and demonstrate what it does essentially. So this is, you can see multiple subunits of this virus, and they all kind of held together by weak interactions, and they're not assembled into anything else. So if we start kind of gently mix them up, and from those bits, it's pretty much, now we see how this simple demonstration, how this co-protein subunits can actually assemble into beautifully arranged symmetrical icosahedral particle that contains nucleic acid. And nucleic acid would promote genuine assembly of these particles on the biological timescale, right? Okay, so Huffington Post picked up on that paper when we published, and they actually wrote this kind of commentary: viruses, nature's self-packing nanoscale suitcases. And the idea was, it was just before Christmas, people going home, and they were saying, well, wouldn't it be nice if our stuff that we need to take home before Christmas would just easily jump into a suitcase. I mean, it'd be amazing, because I'm flying to the US tomorrow, and I'm not packed yet, so I think I would definitely agree with that. But so, okay, well, this is all good. So we kind of work in these fundamental principles of self-assembly. We're talking about protein-protein interactions that drive virus assembly, and RNA-protein interactions. This is also good for several applications. So this was one of the papers kind of first kind of use of this system is that we know that that stem loop, that RNA stem loop that I was referring to, which is present in the genome of this particular virus, we can chemically modify these RNAs, and then we can force this RNA to go into this particle to essentially become part of this nanocontainer. And that's quite convenient, because we can put different functional groups, and I'm not sure this is going to work, and I don't want to use this pointer, but maybe it will work. Actually, it's horrible resolution, but essentially we can decorate particles with RNAs bearing different functional groups, and we can add nanogolds, we can add different fluorophores, or we can put different regulatory RNAs that can be then delivered into cells of interest. And for example, in this case, you can do cell-specific delivery of this bacteriophage-based nanocontainer into tumor cells, or you can use them for diagnostic purposes. So there are quite a few applications of this technique. Okay, so this is something that I did during my PhD. And then, as Margaret pointed out, in my next challenge, I wrote first fellowship on the back of my PhD studies. I wanted to work on something similar, but with a bit more kind of complications to it. And so the virus I chose was the one that actually infects humans. And the virus I chose also had slightly different problem, because this virus has 11 different RNAs. So we're talking about one single RNA molecule. SARS-CoV-2 has one single RNA molecule. Well, it turns out, as I said, viruses are so diverse, they pretty much have every single possible strategy for replication. And David Baltimore, who was another Nobel Prize winner, he came up with this classification system where he basically classified all viruses depending on the replication strategy. And so this particular class of viruses has a huge number of really important pathogens that infect plants, rice dwarf virus here, or they infect a blue tongue virus. There's a recent outbreak of blue tongue virus, deadly virus for cattle in Belgium. Now it's just been reported a few days ago. And there are also, so this is all the viruses, and then rotaviruses. And all of you in this room will have had rotavirus infection at some point in your life. So rotaviruses infect all children, usually under 5 years old. And luckily for us, we have good vaccines, but they don't work that well in developing countries. And just to demonstrate what they do, so Ruth Bishop, who discovered rotaviruses, Australian virologist, 1973, she spotted these beautiful particles under electron microscope, and she noted their appearance that kind of look like a wheel. I mean, to be fair, a lot of viruses do look like a wheel, but the name was rotavirus because they're wheel-like appearing viruses. And they infect pretty much all mammals, right, and birds, and they do infect quite a few different species. Now, the implication of this particular genome organization, if you break up your one single RNA into 11 pieces, is such that when there is co-infection, which happens, and that happens a lot with flu, because flu, just like rotaviruses, also segmented its genome. And flu has 8 segments, this one has 11 segments. When you have co-infection, we have this kind of mixing and matching, and the net result of this is that essentially this virus is going to have something which is akin to sexual process, because you can have different combinations of segments. And that allows it to evolve much faster. So normally what we see in viruses, we have kind of slow accumulation of mutations over time, and that causes what we call antigenic drift. So viruses will devolve slowly by accumulating more and more mutations. Drift implies that it's slow. Here we have something which is called antigenic shift, because shift is much faster. So this is an entirely different process, and it's completely linked to biology, unique biology of this virus. Okay? And so what we're trying to understand in my group, and this is what I've been trying to understand for quite a few years now, is if you have this sort of segmented viral genome, and each of these particles, virus particles, can produce 11 distinct RNA molecules. Each of them are unique, and each of them are required for full infectious particle. How on earth would they recognize each other, assemble? Because this is not cell division apparatus where you have chromosomes and we know more or less how it works. Here we don't have this, and this process happens within hours. So within hours, in the cell, you get these particles. So essentially, I mentioned this kind of unique biology of this virus. This is just a review we recently published, but I just want to highlight several steps, key steps. So one, a virus gets into a cell, and within a few minutes, it will lose the outer shell. Remember, I was referring to this metastability of this virus particle, so it is quite metastable. It will lose the outer protein shell, and that will trigger transcription. So the process when each particle will start spitting out, extruding single-stranded RNA, which acts as a matrix RNA upon which there will be protein produced. And so these different types of RNAs that will produce a bunch of viral proteins, which will then assemble into this sort of so-called early viroplasms. Now, a viroplasm is a term that was introduced or coined by a virologist in the 70s, and probably, or actually not 70s, it could be 80s. And I keep thinking of something ectoplasm, probably because those people are high on Ghostbusters. I'm not entirely sure. But anyway, so first time I mentioned viroplasms term to a physicist, my collaborators in Munich, their question was, what the hell are viroplasms? Why are they called viroplasms? And I said, well, I don't know, to be honest. Frankly, I don't care, because I'm interested in RNA assembly. But it turns out those are really important for us. And so these are the questions we're trying to understand, because eventually this will lead to assembly of virus particle. And if you do electron microscopy, slice of a cell, you can see this kind of budding virus particles off the surface of this viroplasm. So I'm going to give you a much better resolution image of this. This is just to kind of highlight what it looks like. So they're quite dynamic as well. They have really dynamic structures. This is rotavirus-infected cell, or multiple cells rather. And you can see these viroplasms which are tagged with fluorescent protein, with GFP here, green fluorescent protein. And we can drop this compound. And this was literally, for me, was Friday evening experiment in Munich, when one day I came across this paper and I read about this compound called 1,6-hexanediol. And I found it in the lab, and we dropped it into cell culture, and we realized if you prepare 4% solution of this compound and then you drop it into virus-infected cells with these beautiful structures, they immediately disappear. And when you replace it, you replace with fresh medium, fresh cell culture medium where we grow these virus-infected cells, they start immediately reappearing. And you can do this several times. Like, you're too young to remember what lava lamps were, but they're kind of going back in fashion now. So these are things that one of my students, I got one of those kind of lava lamp gifts after I was presenting this several times. We use specific types of microscopy where we can zoom in onto one of these specific droplets, and they're micron size, so really, really tiny things. And we can use light to burn a hole in the middle of this thing, and then you see how quickly they recover. So you can sort of try and bleach a spot of that GFP, and then all that protein just diffuses back in again from the cell. So we knew they're quite dynamic, and which is really fascinating, because we always think of virus assembly as the discrete steps in assembly, but this kind of shows you how dynamic this entire process is, and it has to be. Now we can do something even cooler where I'm just going to show you deep inside the structure. So this is a tomogram. Okay, well it's probably too deep to show you what's going on here, but let me see if I can, so, no, it still doesn't like.
Sorry, can I? Is this a phase separation then?
Yes.
Occurring?
Yeah, I'll go. I just didn't want to call it anything just yet, but okay. No, that's absolutely fine. I did want to bring some vinaigrettes with me, just so a second prop, but I didn't take it with me today. But I'm not sure why it keeps just cutting off, because no, I think you can handle it now, just about. So you can see this kind of, we're doing this tilts of virus-infected cell, and you can see this kind of, the boundary of this inclusion of this viroplasm, and you can see kind of donut-shaped rings. If you kind of squint, and if you believe me, you can see those donut-shaped rings. I'll talk about them in a minute. And then you start seeing this larger granules, which are here, and these are basically budding off virus particles containing nucleic acid. All right? So if I just kind of take you to this still image, you can see even concentric rings of double-stranded RNA bound within each particle, which is, I think, is really fascinating. It's really cool. So I did mention this compound 1,6-hexanediol, which is aliphatic alcohol, and it's known to disrupt weak interactions, similar to those interactions that that drive viral capsid proteins together when they assemble. So they're kind of weak hydrophobic interactions that bring these things together, and we also can use similar compounds to effectively outcompete these interactions, and they also disrupt these droplets, these condensates. The problem with hexanediol is, though, it's quite toxic, so cells usually don't like it, and we don't like it for sure. It's quite toxic. But we did some screen, and we identified a number of compounds and some far smaller, shorter diols. So this one is called propylene glycol, which is generally recognized as safe compound, and it's used, if you look at toothpaste and shampoos, you see propylene glycol is commonly used in those. And so it turns out propylene glycol shown here actually does exactly the same thing. And what's really fascinating, we can also, well, it's maybe not that fascinating when it goes blue. Yeah, it's just giving me a hint that it's time to really wrap it up now. So here you see something else where we can actually look at RNA accumulating in these viral replication factories or viroplasms. And again, when we drop propylene glycol and when we remove propylene glycol, you can see how essentially we wash out all that RNA, viral RNA accumulating within these granules in green. Now, it's pretty much all over the place, and then we remove it and we allow it to reform and reassemble this sort of discrete granules containing viral RNAs, and then you have more granules which are these bona fide viral replication factories. They're foci supporting virus replication. So one of my students here in Cambridge did a number of experiments where effectively, now that we knew that propylene glycol can be used to do exactly the same thing, and the experiment she did, she thought, okay, well, it's far less toxic, so why don't we try and investigate what it does to viral replication without killing cells? And this experiment that she did with propylene glycol, essentially 2% propylene glycol, cells are perfectly happy, they grow well, they grow continuously in the presence of 2% propylene glycol, but when we applied slightly higher concentration at the time when these replication factories are really sensitive to propylene glycol, so they're initially liquid-like and they're sensitive, so they will disappear, then we saw a huge increase in viral titer. So we're measuring particle forming units, so this is how we measure virus titer per milliliter, and we saw this decrease, drop in the virus titer. When we waited longer and then they become resistant to propylene glycol, it doesn't have the same effect. So that was quite a nice finding. I'm not going to go too much into detail, but what's interesting, today we had a paper just accepted with our collaborators. Turns out that propylene glycol has similar effects on multiple viruses, and it also has strong inhibitory effects on respiratory viruses like SARS-CoV-2 and influenza virus. And if you're interested, this paper should appear online soon. But now we started looking at what actually happens within those structures, and so we also started kind of using this top-down approach where we want to reconstitute these things in vitro, because studying these things in vivo in cells, it's fascinating, it's interesting, but there's not much we can do because it's much harder. And now if you remember, I started talking about in vitro reassembly systems where you mix TMV cold protein with TMV RNA. It would have been nice just kind of do this and watch assembly in vitro, and this is always easier. So my second PhD student, Yulia Acker, she did experiments where she started this in vitro reconstitution assays with recombinant proteins. So we make these proteins in E. coli, they're viral proteins, we mix them, and they form these beautiful droplets. So you can actually label one with one fluorophore, the second one with second fluorophore. We mix them together, you see they co-localize, they form these droplets. They're still sensitive to hexandiol, just like in the cell. And you can also see how dynamic they are, so you can see how they fuse. This is without any label. If you can see with that contrast, you see how, well, you could see. I'm not sure I'm going to get this. Maybe. Well, you should have seen there, if you can still see that. No, of course you can't. Just bear with me. No, you certainly don't need to see this. Yes, of course it's not going to do what I wanted to do, but I think it should be here.
Go on, Margaret.
There you go. No, it's all good. No, it's not the same one. Let me just do this then. It really doesn't like me today, does it? No. Oh, give me a second. Yeah, there you go. So, right, I'm just going to skip this one, and I'm going to go on to this slide. And essentially what I want to say with this in vitro reconstitution system is, why is this important for us? Well, first of all, I told you it's much easier to study the systems. The second aspect of this work with doing this in vitro is that we can actually start screening for molecules similar to propylene glycol, but more specific than propylene glycol, because of course we can't use propylene glycol as a drug. But there are other molecules that we can use as antiviral drugs. And this is done in collaboration with Thomas Nose's group over in chemistry. What I'm showing you here, and you don't need to understand the gist of this specific, we call this phase diagram. Essentially anything which is in red here is the concentration regime of these two proteins, an sp5 and sp2, under which these droplets are formed, right? And anything in blue here is when we don't see this droplet formation. And it turns out there are compounds, molecules that would modulate, that would disrupt droplet formation. So this compound here, suramine, 90 micromolar concentration, seems to inhibit formation of these droplets, and 100 micromolar, they're completely gone, so everything is in blue. And just proof of principle, this paper was published a couple of years ago, where respiratory syncytial virus, which is an important virus, again, affecting kids, this compound, rather than disrupting these droplets, it would actually make them less mobile, make them more static, and it turns out that was sufficient to inhibit virus replication, to kill it, because it wouldn't allow these viral replication factories to be as dynamic as they should be, to dynamically support virus replication, which is quite cool. Similar paper was recently published, actually this was work done in Lisbon, Gulbenkian Institute, on influenza virus, and again there's a similar compound that does something similar to influenza virus condensates. And so, I'm not sure why it doesn't. There you go. Yes, actually, yeah, this is Margaret, just, I can see her nodding from behind, not sure. This is something that we published just maybe a couple of weeks ago, recently with Thomas Nose's group. It turns out not only viral condenses can be targeted using this approach. So there are a number of peptides we can use, which are antibacterial peptides, and this is just some of the work that was done with Thomas Naderis and Thomas Knowles, and Margaret was involved in this project here. A number of peptides were identified where they would form similar condensates and inhibit transcription and translation, so protein expression in bacterial cells, and that would effectively kill these bacterial cells. And this is really interesting because we didn't expect this kind of effect, but now we're suggesting there's a new mechanism of targeting not just viral condensates, but also bacterial condensates, and this is really topical in the face of raising antimicrobial resistance. So the last bit I wanted to mention, because I started with the segmented genomes, and I didn't answer the question how on earth we go from 11 different RNAs and multiple proteins into this kind of multi-segmented viral genome. And essentially, we're trying to tackle this problem using bioinformatics, RNA structure probing, a lot of RNA modeling, and biophysics. So we're using this kind of multi-pronged approach and trying to find a way to actually crack this problem. This problem has been around for at least 30 years. Since the discovery of these viruses, people were wondering how these viruses replicate, how they find their segments together. And it's not just rotaviruses, it's also reoviruses with 10 segments. There are fetareoviruses, there are viruses with 9 segments and 10 and 11, all the way up to 12. And so we're trying to understand what the underlying mechanisms are that would allow these viruses to count their segments. And essentially, I'm not going to go into too much detail, but these are kind of a few things why we care about this question. Well, first of all, if we know what the rules are, we can start introducing foreign genes into this virus. And I know it sounds scary because we know gain-of-function studies in virology, they're kind of controversial, but we need to understand how these viruses work and whether we can make new generation vaccines based on rotaviruses, because they have great potential for introducing foreign genes. They can be dual vaccines against norovirus and rotavirus, for example, or calicivirus and rotavirus. And some of the work we did even on combined vaccine, rotavirus, SARS coronavirus, because SARS-CoV-2 can also replicate in intestinal tissues in children, for example. So again, if we improve this virus, if we understand the underlying principles, we can make them replicate to higher titers. So we'll be getting viruses that replicate better, which means we can make better vaccines, because we can't use anything that doesn't replicate well as a good vaccine candidate. It has to be a viable vaccine candidate. It has to grow to reasonably high titers, otherwise it won't be a viable vaccine candidate. And so 2 talented postdocs in my group, bioinformatician Daniel and Lee, who unfortunately left now is Glasgow MRC Virology Unit, they did a lot of work trying to crack this kind of RNA interactome. So you have all 11 segments, for example, and we're trying to work out how these segments talk to each other. What is the RNA code that allows all 11 RNAs to talk to each other, which would effectively ensure that all 11 RNAs are co-packaged into one particle. So just another publication that we had a couple of years ago on this combination of rotavirus and SARS-CoV-2, where we can deliver SARS-CoV-2 antigens using rotaviruses, for example, these recombinant rotaviruses. And I think this is it, pretty much. I'm not going to dwell on other things. I'm just staring at this because I can't really see what's actually happening on my slides. And yes, okay, well, that's obviously our group and my great collaborators and funders. We do have several other projects that I didn't have time to talk about today. Like I said, we work on RNA therapeutics. This is a project funded by Pfizer. We have a number of projects where we're working on condensates, biomolecular condensates, as I mentioned. We're interested in RNA biology of rotaviruses. We're also interested in fundamental biology of rotaviruses. A lot of this work was done by my new group members and older group members. Some of them moved on. And Catherine Lilly here, who's my close collaborator with whom we do work on the Pfizer project. And my group here, which I think I mentioned pretty much everyone in my group. So Daniel, who was there, Yulia, she sat next to me. I should really update some of these photos. They kind of just like viral plasms that are dynamic, my group is also dynamic. People come and go. Some of the students who've done some work, unfortunately they're not here, but I think I was quite fortunate to have really good students in my group, and just kind of nice to work in this environment where people discuss all these ideas and care about the science that we do. And yes, with that, I'd like to thank you again for your invitation, and thank you for just being with me here tonight.
Thank you very much for the talk. So, you're okay to take questions.
If there are any questions.
Thank you for your talk. So, my understanding of like antibiotics is like they have obviously beneficial applications, but they put out things like our gut microbiome. Obviously, viruses are also a component of the microbiome. So, with these antiviral drugs that you're developing, have you considered the consequences of them, considering the beneficial applications of viruses in our bodies?
Yes, so beneficial applications of viruses. It's kind of loads of questions. So now I'm just, for some reason, I'm thinking about applications of phage therapy, for example, viruses that infect bacteria, and that phage therapy has been a hot area of research, especially 10, 20 years ago, and it still is for topical applications, because delivering phages to treat bacterial infections is not that easy, because phage itself will trigger immune response. But for topical applications, it's still, I think there's a lot of research in this area. Another issue is they're highly specific to bacteria. So it's that kind of broad specificity, which is what we gain from using antibiotics, we lose that with phages. But again, because of antibacterial resistance, antimicrobial resistance, I think any tool will be useful, including phage therapy. Now, what was the second question again?
That was only really one question, but it was more, what are the beneficial roles of viruses within the body? And if you have these generic sort of antiviral drugs, would they be implicating that process?
In the process of developing resistance?
Not just resistance, but if there is like a beneficial role of viruses to play in the microbiome
Okay.
within the body.
Yeah, I mean, it's an interesting question. There are several hypotheses about some of chronic viral infections that may play a role in immune modulation. One of the classics is herpes virus infection, because most of us would have herpes viral infection at some point of our life. I'm talking not just herpes. There's human herpes virus type 1, there's type 2, there's cytomegalovirus, which is CMV, that's type 3, and EBV, which is Epstein-Barr virus. There are quite a few herpes viruses, they all kind of belong to the same family. So most of you in this room will have had some antibodies against one of these viruses. And they may play a role in some sort of immunomodulation. We kind of co-evolved to exist with these viruses. They don't necessarily actively replicate. They infect us at some point of our lives and then just stay dormant up until they get reactivated. But this is all kind of quite speculative, because there aren't any robust studies that would say they are important. We haven't done kind of studies where it would completely keep people pathogen-free for the entirety of their lives and then say, yeah, okay, they definitely benefit from these chronic infections. Yeah.
Great talk, Alex. So this might be a naive question, because I don't know anything about it, but what actually moves these viral plasms around in a cell? Because the movement looks quite erratic from the movies you have shown. And then the second question is, how did you manage to get this FRAP experiment if they actually move so much? Because they were standing still for a minute or so, right?
Yeah, that's a good question. So the first question is, how do they move? So there are lots of things that play roles. So we know if we use different inhibitors of cytoskeletal protein assembly, either block tubulin polymerization or we block actin filamentation, it will pretty much become quite static, so they don't move as much. So clearly there's a lot of motion that happens in the cell, and they become quite mobile. There is also the smaller ones, presumably just Brownian motion. There's also that, and we can't really exclude that for the smaller ones. Now, second question with FRAP, it's not trivial. It's hard, and people who do FRAP on mobile things, they, so in this case, it was a custom-built setup where we do wide-field microscopy, and we use very low power illumination, so we don't bleach them, so FRAP, for those who don't know, essentially based on recovery of a fluorescent signal after photo-bleaching. And we shine a really powerful laser, and you have to shine it right in the middle of this spot, so then you can start watching how, over time, due to diffusion or mixing of these fluorescent molecules, you start seeing full recovery. And that gives you some information about how quickly molecules move in and out. And that way you know that it's more liquid-like rather than static. Yes, I mean, for really tiny ones, it's really hard. For larger ones like this one, which is about 2-3 micron size and larger, you can. In terms of how quantitative it will be, yes, I don't put any numbers in this because it's hard. Yeah.
Is it on? So thank you, Alex. It's a lovely talk. When you were looking at the dials, my impression was that they act generically on many different systems, and then when you go to your when you're looking at your phase separation screen and you're identifying molecules that inhibit that process, they're much larger molecules, and I assume. So my question really is does that mean that they're specific to those particular viruses, and is anything known about how they act? Do they bind? Do they just affect phase separation, but don't? How do they work?
This is this is a great question, Sophie. So dials, they're definitely non-specific. So we've seen that, and people have reported from many systems that form these biomolecular condensates, as long as they're kept by probably weak hydrophobic van der Waals interactions. If they're mostly held together by ionic interactions, there are some condensates, DNA, RNA, protein condensates, then quite often those dials actually don't work on them. But high salt would in vitro. And you do need quite high concentration. So when I'm showing, I know it's kind of counterintuitive, but for someone who's not a chemist, if you say 2% propylene glycol, it sounds, well, it's nothing. But actually, if you start talking in terms of molar concentration, it's quite a lot. Now, for those who don't think about water concentration in cells, it's 55.5 molars, a lot of water around, and actually still the bulk of solvents in biology is water. So now with those small compounds that I showed, and not just from the screen, but also the RSV paper, they knew that compound binds one of viral proteins specifically, and it just prevents phase separation, presumably by binding to one of those proteins and blocking one of the interaction sites. There is an interesting theory amongst biophysics of condensates, and people call it stickers and spacers model. So essentially, you have stickers which, in this case, we're talking about these little magnets that will hold things together, and then also spacers, which is space between those magnets. And essentially, stickers will bring multiple molecules together, and spacers are they're needed because you still need some sort of freedom for these molecules to move around with respect to each other. And that's what creates this kind of condensate, which is not like a sturdy kind of rigid condensate, but more mobile. And so those compounds can bind to these sticker points with high affinity and then prevent or kind of either stabilize, make them even more stable, or weaken those interactions. And in that case, it will inhibit condensate formation. So I think the point where people quite often they make an argument saying, well, you can't target condensates because it's all weak and non-specific, so it's just not going to work. Well, my argument will be, well, we have quite a few examples where it does work, and it works exactly by specifically targeting components of this condensate, and that prevents condensation. So, yeah.
But the condensates themselves have to have some specificity, right?
Yeah.
Otherwise, there'd be all kinds of stuff in there.
Yeah, no, what I didn't show, this is a really good point actually. So in this viral condensate, you only find viral RNA, you don't find non-viral RNA, which again biologically makes sense. So some of the data didn't show, but we did RNA single molecule fish where we're looking at all the RNAs that go in there, and we didn't see any non-viral RNA. And we've done some interesting work where we showed that specificity is imparted by RNA binding protein, which is viral polymerase, which binds, again, specifically to viral RNA. Just like, remember when I showed MS2, where you have this stem loop that binds its cognate protein. Here you have polymerase that binds its cognate RNA, and that becomes part of that condensate. So there is specificity. But there are lots of types of condensates, and they all show something I didn't show you today. We actually did experiments where we can reconstitute another type of condensate called stress granule and this viral condensate, and we can use an instrument, optical tweezers, where we can bring those together, smash them. They're both liquid-like, and then they fuse, but they still stay separate. So you have stress granule condensates in the middle, surrounded by viral condensate, but they don't mix. Probably dig out this image, but I can show you later.
Hi, thanks for the talk. Sorry, if I'm not mistaken, when you were talking about sort of a vaccine for rotavirus and the high titers and the gene stuff, is that a live attenuated vaccine? And sort of on that, is there any reason for rotavirus you might expect that to be more successful than, say, like a protein subunit approach, the mRNA sort of approach, or any of the other vaccine types?
Yeah, so I recently had a conversation about mRNA vaccines with some skeptics of mRNA vaccines, hardcore virologists, and they said, well, it's going to be a massive failure. Of course, just a matter of time, because we needed a quick fix, and investors, they jumped on this bandwagon because they saw the potential of mRNA vaccines, but actually they're probably not as good as we think they are. And there are better platforms for antigen delivery than just mRNA vaccines. They're versatile, they're flexible, you can quickly ramp up production, they're great in that respect, but they're probably not really a solution for making everyone immune. So there are still many questions about in vaccinology for sure. And we know newer generation, those subunit vaccines you're referring to, they seem to be more immunogenic. They boost immunity, mucosal immunity, for example. So again, it protects you against reinfection rather than just being protective like some of the mRNA vaccines. So clearly, attenuated viral vaccines are still highly efficient. We know that we have quite a few examples of those kind of old generation attenuated vaccines that work really well. What we don't know, and I think it's specific to each virus and each pathogen, some infections will essentially provide really good, robust immune response, and that will protect you against, like, one classical example is chickenpox. And some just don't. I mean, we get reinfections all the time, and it's not just because there are multiple strains and variants, it's just simply there's something else about biology of this pathogen. And using the whole organism, whole virus, the whole bacteria attenuated, it can actually give us kind of this bit of a leeway, so we don't have to target one specific product, we're targeting multiple things, and it can provide better immunity. I don't want to say also disparage the mRNA vaccines, of course, so please don't quote me on that. I mean, they've been incredibly efficient, they've been very successful, but and there are more vaccines coming up, especially in cancer biology therapies. Using mRNA vaccines is a very powerful approach, but there are still disadvantages. They're still not perfect.
Anyone else have any questions? I was just wondering myself. You showed how how you can induce phase separation inside with viral RNA. I'm just wondering if there's any way you can deliver that in a biological system, say, use it as a drug. Are there any efforts to look at that?
Do you mean delivering condensates into cells?
As in inducing
Inducing condensates. Yeah, people exploring that. It's quite interesting approach. There have been quite a few reviews written on this. There are groups in pharmacology working, Janet Kumita, Laura Itzhaki, they're working on sort of evolved proteins and RNAs that can assemble into condensates. They might not be the most efficient just yet, but there are lots of kind of general principles that we understand how to essentially make them work and how to assemble. And the idea is, I mean, you can think of a few applications. One that immediately comes to my mind will be try and have sort of control system where you can capture mRNA in a cell and then release when it's needed, because that's essentially some of the main roles of these condensates in cells. If you're talking about P granules, for example, involved in development, they store mRNAs when they're not needed, but at some point of development, they will dissolve quickly and release these mRNAs available for translation. So you can have that sort of evolved system where it can be useful, can be quite potent.
Thank you very much.
All right, thank you.
So, yeah, if no one else has any questions, then yeah, thank you.
Thank you very much.
Feel free to hang around next door for more refreshments.
Keeps me from rankers.